Hi, this is Ahmet Danli from Cold Spring Harbor Lab and Genome Research. This week we have the 86th Cold Spring Harbor Symposium on Genome Stability and Integrity. Joining me today is Scott Keeney from the Sloan Kettering in New York City. Thanks for being here and welcome back to Cold Spring Harbor.
My pleasure.
Before we get to your research, I'd like to ask about your background, where you were born, what inspired you to become a scientist, and how you ended up being interested in meiosis.
Okay. I grew up in Baltimore, so down the coast. I started out in engineering and then briefly pre-med and in preparation for med school. I went to undergrad at Virginia Tech, and in preparation for med school, guidance counselor suggested that I get some research experience. And so I asked my biochemistry professor if I could work in his lab. He said sure, and pretty much first day I knew that was what I wanted to do. So I was a lab rat throughout college, and then kind of got the bug to travel to the West Coast. So I applied to graduate schools mostly on the West Coast and ended up choosing to go to Berkeley. And once I was at Berkeley, first person I rotated with suggested that I work on the side of his lab that worked on DNA repair. This was Stuart Lynn, and that kind of cemented my love of chromosomes and DNA damage and repair.
Nobody can blame you. It's very interesting.
Yeah.
So we're chatting before your talk. I know that you work on regulation of double-strand breaks in a couple of organisms using different multi-pronged approaches, and I do not know what you're going to talk about tomorrow, so would you mind giving us a preview?
Sure. So my lab studies homologous recombination during meiosis. So this process is required for the maternal and paternal chromosomes to pair with one another. The process of recombination starts with double-strand breaks that the cell makes in a developmentally programmed way, and that relies on the protein SPO11, which is the catalytic center of the DNA breaking machine. So I've been studying SPO11 my entire independent career, which is almost 25 years now, and I started out when I started my lab at Sloan Kettering wanting to work on it at all levels, biochemically, genetically, cell biological approaches. And the genetic approaches were working, but the biochemistry never did. And so I spent a lot of time at the beginning trying to get SPO11 purified, to purify its partner proteins that we knew about, and just failed dismally. And fast forward to a number of years ago, maybe about 5-6 years ago, a postdoc in the lab, Corentin Kleis-Buart, who was a really talented biochemist, decided to try again to tackle what we had failed to do for many, many years. And in the intervening years, we had learned a lot about
what
SPO11.
not to do.
Sorry?
What not to do.
Yeah, what not to do. We learned a lot about what proteins interacted with what proteins, and in very short order, he was able to purify recombinant complexes of SPO11 with some of its partners and some other proteins that are also important for SPO11 activity but are not part of a stoichiometric complex. And people, sometimes ask me, well, what was the key to success? And like it usually is, it was the right person. So because he was very talented, is still very talented as a biochemist, he was able to succeed where nobody else had been able to before that. So that's the backdrop. So he has since left and started his own lab. He's in Belgium.
Okay.
And I am going to be talking today about some of his work as sort of a backdrop, and then talk about unpublished data from people that have joined the lab since then. A postdoc, in particular, Kai-Xian Liu, and graduate student Zach Zhang. and I'll be presenting a cryo-EM structure of SPO11 bound to DNA and some single molecule studies of DNA binding activities of some of the partners of SPO11.
I'm looking forward to it. So, just from your research in general, the number of double-strand breaks in a mouse cell, I think it's in the order of a few hundred, I guess, right? Is that proportional to the genome size?
No, that's actually a really interesting point. So, mice, as you say, make something like 200 to 300 double-strand breaks. Yeast actually make a fairly similar number, even though their genome size is much smaller. So they make about 150 to 200 breaks per meiosis. In contrast, C. elegans and Drosophila make only a couple dozen breaks.
Oh, really? I didn't know.
Even though their genome size is intermediate.
So the obvious question is, do we know why?
So the why we know, the how we know less about. The why has to do with what the purpose of recombination is. So in many organisms, including whatever plants have been studied at this level, mammalian cells and in yeast, recombination serves sort of two cellular functions. One function is to provide a mechanism for DNA molecules to search for other DNA molecules that are homologous. And so it actually allows, the recombination process actually allows the maternal and paternal chromosomes to recognize one another as homologs. The second purpose is a subset of those double-strand breaks gets repaired in the form of a crossover recombination product. And that crossover, along with sister chromatic cohesion, makes a temporary connection between the homologous chromosomes that allows them to orient stably on the metaphase 1 spindle and then be pulled apart when the cohesin is dissolved. And so those two functions, pairing and the temporary connection to stabilize on the metaphase plate, require that there be many recombination reactions occurring simultaneously all along the lengths of the chromosomes. In Drosophila and C. elegans, the second function is still there. The crossovers are still necessary to stably connect the homologous chromosomes, but the pairing function doesn't require recombination. Those species have evolved different ways for homologous chromosomes to recognize one another that don't involve recombination. And so as a result, they make fewer breaks because they only need enough breaks to make the crossovers. They don't need all the extra breaks to do the pairing reactions. So that's sort of the why for why different organisms have such vastly different numbers.
So there is quite some difference, sounds like. What about the proteins involved? Are they highly conserved or not necessarily?
So that's another really interesting question that we're actively studying right now, and there's a postdoc from my lab who's here at the meeting, gave a poster on this. In general, meiotic processes are conserved in sort of the broad strokes, but there are a lot of really big differences between species when you actually drill down into the details. And specific to your question, the SPO11 and its partners are some of the fastest evolving proteins in the genomes of species where they exist. So SPO11 itself is universal or near universal in eukaryotes. There are some, it looks like dictyostelium and other social amoeba don't have SPO11, but otherwise every other eukaryote, as far as we know, has SPO11. And its partners are, many of its partners are found in other species, but in some cases their amino acid sequences have changed to the point where they're really hard to see. We now know that most of them are in fact conserved, but they're evolving really fast, and we're trying to understand what the causes and consequences of that very quick evolution are. And short answer is we don't know why they're evolving so fast.
So I think this is a fascinating topic. So some naive questions potentially. So what happens with the X and Y?
That we could talk about for the next hour if you want.
Okay. I think we have a little bit less than that, but Or, small chromosomes, like things where the size seems to be limiting.
Mm-hmm. So for the X and Y in spermatocyte meiosis, in mammals, in most mammals, mice, humans, the X and Y still have to pair with one another and recombine, but of course they're not homologous over most of their lengths. And so they rely on a very short segment of DNA called the pseudo-autosomal region at the distal tip. And in mice, that's only about 700 kilobases. So that tiny section of the genome has to get a double-strand break. That double-strand break has to support the pairing reaction for these chromosomes to search for one another in the nucleus. They have to pair with one another, finish the recombination reaction, and make a crossover. And then that crossover has to stably remain cohesed until anaphase 1. And so there's a lot of challenges for the X and Y, and that's one of the reasons why XY missegregation is the most common type of whole chromosome aneuploidy in male meiosis in mammals.
Well, I used to do biochemistry and also work on transposable elements and small RNA silencing. So inevitably, I'm tempted to ask about transposons and how they fit into the picture, because it's one of those things. They are quite important, but there have been many technical challenges. So sometimes when you talk to people, they discard the transposable element reads when you need to, but in your case, they might be relevant probably.
Yeah.
But also technically challenging. I was just curious, how do transposons fit into the picture here?
They're a really important part of this. So something I say to the students in the lab is, if you think about the dynamics between transposons and their hosts, transposons can jump around in your eyeballs all they want, but that's not going to do anything for their propagation. Mm-hmm. It's only if they mobilize in the germline that it's going to matter. And so the germline is the battleground between transposable elements and the host. And there are a lot of very interesting host defenses against transposons that are germline specific. It's actually kind of circling back to this question of why SPO11 and its friends are diverging rapidly. Rapid sequence diversification of proteins is often a sign that there's a genomic conflict of some sort. And so one speculation is that it is actually conflicts between either host and transposons or host and meiotic drivers, which are other kinds of selfish genetic elements, that those might actually be the driving force behind these rapid evolutionary changes. So I think transposons are really important. We've done a few studies over the years looking at so SPO11 cuts across the genome, but it cuts very non-randomly. It prefers to cut in certain locations. And so in both yeast and in mice, we've been very interested in breaks within repetitive DNA. So as you mentioned, it is a big challenge to map sequencing reads to the repetitive part of the genome, but that's where I think a lot of the interesting biology is, and I'm clearly preaching to the choir here on that. So we're very interested in improving the tools for doing that mapping and trying to understand what happens when breaks are formed in repetitive DNA.
What about polyploidy? Does it bring its set of challenges, and whether we can learn anything from them regarding aneuploidy or whole genome duplications?
So this is not something we actively study, but it is something that is kind of at the forefront of research in the community. And there have been some interesting studies recently about how polyploidy affects the recombination and chromosome segregation process, particularly work from Kirsten Bombley's lab in Switzerland, where they have identified that polyploidy creates new challenges for chromosome segregation, because ultimately the homologous chromosomes have to join together in pairs and separate. And if you've got say, a tetraploid, where instead of a pair, you have two pairs, and all four are identical. If they don't connect 2 by 2, but instead 3 of them connect and 1 doesn't, or all 4 get connected, that would be a tangle that the meiotic spindle can't pull apart. And so that extra ploidy creates a challenge for the cell to not just match chromosomes based on homology, but to do it in a 2 by 2 fashion. And they've identified some pathways that cells seem to have evolved, that stable tetraploid species have evolved to deal with those challenges. And it's another example of how the meiotic process is quite plastic evolutionarily, because the chromosome constitution really affects how the recombination process has to work.
Do the new long-read driven technology-driven structural variation data, does it give you further testable hypotheses in your system with respect to how they might be generated or propagated? And
yes, we have not yet really kind of gotten on the bandwagon for the long-read technology, but it is very important for understanding, again, kind of coming back to this issue of recombination initiated in repetitive sequences. If you make a break in a sequence that has a copy just at the allelic position on the homolog, that's your only choice. You either the sister chromatid or the homolog. But if it's a repetitive sequence, then you have many choices, and that recombination can give rise to gross chromosomal rearrangements, deletions, inversions. And so the long-read sequencing technology, one way that's important for us is it actually gives us a better view of what the genome looks like. Because if we do, any kind of mapping technology to map where double-strand breaks or recombination events are happening, the maps we get are only as good as the reference genome we're mapping against.
Right. So we're seeing a lot clearer, I guess, is the point. So in the last few minutes, I mean, we could talk about meiosis, all day long, but I think we need to limit our time. I wanted to ask you about science and society. And part of the reason why I do this is even though the pace of scientific discovery is, breathtaking in some fields, the science communication and the value given to science and society is really lagging. So I think it's our responsibility as scientists to communicate our science better, why we do it, and this is also necessary for, generating the diversity that is fair and necessary, as well as the funding that's necessary. Mm-hmm. So if you have anything to say about that, the floor is yours.
I think maybe I'll just take the diversity angle on this. I think it's very clear that having diverse perspectives is better for just the healthy functioning of science as a community exercise. There's lots of examples where fields have stagnated because of siloed thinking and a lack of diversity of ideas. And I think that goes across the board, not just for intellectual diversity, meaning physicists and chemists and biologists talking together, but also for diversity of background, and particularly in health sciences, where it becomes clear that a lack of diversity at the level of the researchers and the clinicians plays out directly in a lack of equitable health outcomes for communities. But also, another angle that I like to mention is in kind of today's climate, this is the beginning of Pride Month, and I think what we're seeing in the United States is a really strong backlash against LGBTQ rights, but with a particular focus on trans people. And I think that's something that, so I'm gay, I've been openly gay for essentially all of my career, and I think it's something that's much more challenging for trans people to navigate life than it is for cis gay people like myself. And I think that all of us need to be aware of the particular challenges that our trans colleagues face, and especially now where the environment is become so toxic for trans rights, and it's very distressing to see all these laws being propagated in different parts of the country that they serve no rational purpose other than to empower an anti-trans bigotry.
Well, I hope they don't have to wait as long to get the rights they deserve.
Yeah, I hope so.
All right. On that note, thank you very much. It was a pleasure talking to you.
Thank you.
And see you at the next meeting, I guess.
Yeah, thanks.
Thanks.